All right, so I don't see any other questions. So I suggest to move if there's none. And by the way, panelists, feel free to unmute and ask questions too if you want. You're allowed to, don't worry. So we can move to the next presenters. And we have both Michael Sutton, Dr. Michael Sutton, as well as Dr. Iwase, Shigeki Iwase. I hope I pronounced your name correctly. So I'll start with Dr. Sutton. He's a professor of the Department of Molecular and Integrative Physiology and senior investigator in the Michigan Neuroscience Institute in the University of Michigan School of Medicine. And the research program in his lab focuses primarily on the establishment and refinement of synaptic connections in the developing brain and remodeling of those connections by activity in mature neural circuits. His work has been published in top scientific journals. Dr. Sutton enjoys a close collaboration with Dr. Shikage Iwase's research group in the Department of Human Genetics in Michigan. Both of them are here tonight. So together, their laboratories have launched an intensive effort to understand how chromatin remodeling contributes to neurodevelopment disorders. A significant aspect of the initiative is aimed at understanding how loss to the gene RAI alter neurons, synapses, and circuits in SMS. Their goal is to contribute to developing novel therapeutic strategies to treat SMS based on a deeper understanding of the cellular roles of RAI in the brain. With a brief introduction, I would like then Mike and Shigeki present.
I can only make one of you a host. Who would you like it to be?
Make me a host if you don't mind.
Yes.
We think maybe I should speak rather than us going every other word.
Okay. And once I make you host, if you wouldn't mind, if this stops recording, if you would start recording, please.
Sure
and I'll
will.
get recording from you later.
Yep. Okay, looks like we're still good.
It looks like we're still recording.
All right, wonderful. Okay. Let's get set up here. Still recording? Wonderful. Well, thank you very much for that wonderful introduction, Gianluca. It's wonderful to meet you. Thank you, ladies and gentlemen, for being here. I'm absolutely thrilled and excited to present some of our recent work on RAA1 and how we're thinking about how this work might lead to some novel therapeutic strategies for SMS. So let me start by sort of introducing you to the team. These are the people that do all the work in the lab. These are the folks in both my lab and Dr. Wase's lab who are focused on RAA1 or SMS. And they're a really talented group. They're really devoted to this work. They work very hard to try to get a deeper understanding of what RAA1's function in neurons is. And so we're really indebted to them. And I should say that all of the work that I'm talking about is a partnership between my lab and Dr. Shigeki Iwase's lab in the Department of Human Genetics at University of Michigan Medical School. We're grateful for extramural funding from the National Institute of Health. This really helps support our broader interests in chromatin regulation, but we're especially grateful for support from the SMS Research Foundation, because this has allowed us to ask very targeted questions about RAA1 function and how that might relate to SMS disease progression. So the primary goal of our work is to advance the development of therapies for SMS based on a better understanding of the physiological roles for RAA1. We know that loss of RAA1 has pretty severe effects for brain development, but we don't really know a whole lot about what RAA1's roles in neurons are during normal development. And so a big goal of our work is to try and get a better understanding of that. So just in some context, you know that neurons, like all cells, have specialized functions, and this is because they express a unique complement of proteins. They express ion channels to make them excitable. They express neurotransmitters and receptors to allow them to communicate with other neurons. These specialized proteins, the blueprints for these, of course, are in the nucleus in the form of the DNA genetic code. These genes can be read out and copies made into a molecule called RNAi through a process called transcription. These RNA molecules have these coding regions, but also these intronic regions that are non-coding. These intronic regions get spliced out, and then these mature mRNAs get exported into the cytoplasm. the rest of the cell, where they serve as a blueprint to make these proteins. So what we do know about RAI1's function is that it seems to function here. Seems to function at the level of DNA and regulating the expression of various genes. Although RAI1 doesn't bind DNA directly, it does bind a complex of proteins associated with DNA that we call chromatin. These are histone proteins, and the DNA wraps around these histone proteins, and RAI1 seems to recognize this complex and thereby regulate gene expression. So given this, we want to have a deeper understanding of RAI1's function. And so to get at this, we want to profile the genes that are direct targets of RAI1. And so to do this, we take advantage of the fact that during transcription, the DNA bases in DNA get copied into an mRNA template, but thymine, the normal base for DNA, gets replaced with uracil in mRNA. So we can take advantage of that and give cells a different form of uracil called bromouridine that they then take up and incorporate into their mRNA copies. This allows us to sort of tag those newly made mRNA transcripts as the cell's making them. So we can ask, when we acutely remove RAI1 from cells, what are the consequences in terms of the genes that are being made? How does it directly regulate gene transcription? And so to get a better understanding of this, we want to study this in an additional context. We also want to know, are these RAI1 dependent genes, which we think are important possibly for disease progression, are they activity responsive or not? And so, as you know, neural activity is very important for neural circuits, but it's very important that neural activity be stable. And because of all the synaptic connections between neurons, we know that that activity has the propensity to become unstable very quickly. You can have persistent increases in activity, like seizures, but even things that are pre-seizure that compromise circuit function. But you can also have too little activity. So circuits have to maintain this stable level of activity. And how do they do this? Well, fortunately, neurons are endowed with a safety net. These are processes that kick in when activity gets too high or too low. And so when activity gets persistently high, a process kicks in first at the level of transcription that basically weakens the connections between neurons to allow this firing rate to come back into this stable range. So the signature of this event then is this downscaling of synaptic function in order to bring neural activity back into this stable range. Likewise, when activity gets too low, neurons upscale their synapses. They increase the strength of their synapses in order to bring activity back into the stable range. We know these processes are very important, particularly in developing neural circuits, for maintaining stability and allowing these circuits to function properly, even though there's large-scale synaptic remodeling that's going on. It's also important, however, that these mechanisms don't get turned on when they're not supposed to. So you wouldn't want to turn on this upscaling program, for example, when activity is stable, because this might push you into an unstable, hyperactive state. And as we heard from the first talk today, there is some evidence that neurons are in this hyperactive state, at least in some regions of the brain. So to profile new genes, we took advantage of this Bruce-seq approach. We took mouse brains and we prepared neurons from them, allowed them to mature in cell culture over a few weeks, and then we disrupted activity. We either suppressed activity with an agent called prototoxin. This blocks the ability of the neurons to fire action potentials, or we took advantage of this agent bicuculin, which basically increases activity in the network by decreasing inhibition. Okay, so neurons are either persistently activity suppressed or persistently overactivated. We then profiled all these new mRNAs and asked how these states changed the transcriptional gene expression programs that the cell was engaging in. And what we found actually is that most of the genes that were turned on by activity suppression were actually not turned on during activity overactivation. So the genes were actually quite separated. So what this told us is that the gene expression programs that underlie synaptic downscaling are really separate from the gene expression programs that underlie synaptic upscaling. And so why am I telling you this? Well, the reason I'm telling you this is because we think this might really relate to something that RAI1 does in neurons. And so we got at this by first acutely removing RAY1 from neurons. We did this using a technique called RNA interference, where we could deplete RAY1 from neurons pretty acutely within about 24 hours. And we achieve about 80 to 90% loss of RAY1. So it's acute. So we can ask what happens now to gene expression when we do this. And what we find is that there's a variety of genes that are up or down regulated. Most of the genes we find that are regulated are down regulated, which is consistent with the idea that RAY1 is generally promoting the transcription of its targets. But also we find a significant cohort that are upregulated, suggesting that under some circumstances, RA1 might actually inhibit a transcription of its target genes. We found a couple features of this data set. I won't go into all of it, obviously, because the data set's complex, but we found some important features. One is that these RAY1 dependent genes, RA1 dependent genes, are enriched for synaptic proteins and regulators, suggesting that one thing that RA1 regulates are things that are important for how neurons are communicating with each other. The other big hit we found were circadian genes, which is not surprising given the disturbances in circadian rhythms and sleep-wake cycles that we see in the SMS population. So we're already sort of following up on some of these by looking at some of these downstream genes and seeing how they regulate synaptic function and synaptic phenotypes with RA1 depletion. The thing that we noticed that was very surprising is that the overwhelming majority of these genes were TTX response genes, genes that are turned on when activity is suppressed. This surprised us, and we see this at the level of the genes that are statistically regulated, the big hits, but we see this in the overall level of the transcription well. If we look and see that genes that are basically turned on by TTX, by activity suppression, generally go up when RAY1 is depleted, and genes that are turned off generally go down when RAY1 is depleted. So there seems to be a tight correlation between these genes. We don't see that correlation when we hyperactivate the neurons, suggesting that RAY1's really targeting genes that are turned on by activity suppression, the upscaling transcriptional programs. Curiously, when we actually look at this, when we suppress activity, we find that RAY1's ability to regulate gene expression is mostly lost. This suggests that it's really functioning when neural activity is stable in terms of regulating gene expression. So we used a different approach called ChIP-seq to follow up on this and to ask another question, which is, how does RAY1 bind its target genes? So can we identify true RAY1 targets, RA1 targets, by virtue of their changes in expression, but also a direct binding of RA1? And so we asked, are the genes that we identified in our earlier data, are they directly bound by RA1? And how does activity regulate this binding? And to summarize sort of a large data set and woefully small number of data panels, I just say that we do see that RA1 does bind lots of genes throughout the genome. It tends to bind in gene regulatory elements called promoters, which are the ones that are most important for regulating expression. So this is all consistent with the idea that RA1 is acting as a direct regulator of expression of those genes. The surprising thing, again, is that when we shifted activity into either inactive or overactive states, we found that almost all of this binding was gone, suggesting that changes in activity are causing RA1 to dissociate en masse from chromatin and no longer regulate its target genes. Again, most of the RA1 targets that are bound by RA1 are TTX response genes, showing that it has at least a preference for these genes that are turned on during synaptic upscaling. So what does this all mean? So we think that actually what it means is that RA1 has a very unique role in controlling the expression of these transcription programs that lead to these adaptive synaptic changes. So specifically, it plays a role in suppressing the synaptic upscaling that you would see when neural activity is stable. So under normal conditions, when neural activity is stable, you don't want to have these upscaling gene expression programs because that's going to push you into this unstable hyperactive state. But when you lose RA1, you lose that checkpoint. And now, even during stable patterns of activity, you may get into this unstable state. And so we think that that's actually very, very important. So when activity shifts over either to a lower state or to a higher state, we see massive loss of RAY1 from chromatin, showing that it's dissociating during these different states. So how do we test this model? So one thing we can do is measure the synaptic currents and ask, are these, if we deplete neurons of RA1, do we see that the neurons upscale? And in fact, they do. So using a technique called electrophysiology, this whole cell patch clamp electrophysiology, we can measure these synaptic currents. So these are measuring the release of a vesicle that contains neurotransmitter. In this case, it's glutamate. It binds to postsynaptic receptors. It opens up an ion channel and passes a small current, and we can measure that. And we see that these currents actually get bigger when we deplete neurons of RA1. We put RA1 back. We can rescue this or reverse the currents back to normal. And the pattern of this increase is very reminiscent of what you see with activity suppression, in that all of the synaptic currents seem to get shifted and bigger, not just a select few. So this has, which is sort of a signature of synaptic upscaling. So together, this data suggests that one novel functional role for RA1 is to regulate this, the induction of the synaptic upscaling event, particularly when it's not appropriate for it to be induced during periods of stable patterns of activity. So to follow up on this, we're really interested in whether these roles are actually found in human neurons. So is this something that's only unique to the mouse? Or can we see these same gene expression targets, the same functional role for RA1 in human neurons? And so to address this, we've taken a cellular reprogramming approach. We've taken human embryonic stem cells, used a CRISPR gene editing approach to remove either one or both copies of RA1, and then differentiated these cells into neurons, allowed these neurons to mature and form synaptic connections so that we can then measure some of these functional effects, as well as the genes that RA1 is targeting in these different circumstances. So I'm saying we're doing large-scale profiling at all these different stages of neural differentiation. We're still acquiring the data, so I don't have any of it to show you. But this includes sort of morphological analyses, as well as studying the genes, how the gene expression changes at these different stages, to get a better sense of how RA1 might be regulating neural development. But what we can actually study is test this idea of whether loss of RA1 leads to synaptic upscaling in human neurons. So in the neurons that we derive from human embryonic stem cells, loss of a single copy of RA1 does indeed lead to this upscaling effect. So we see that the synaptic currents scale up, and to a lesser extent, we see the same effect in the knockout where both copies are gone. And again, the way that this shifts, it seems like all the currents shift and get bigger, which is a characteristic signature of what we see in synaptic upscaling. So some of our ongoing projects that are inspired by this work are to, one of the main goals is to identify the genes in human neurons that are directly regulated by RA1 and are conserved. We think this is important for identifying novel molecular pathways as therapeutic targets. And this will allow us to test if modifying those pathways impacts these synaptic and behavioral effects of RA1 deficiency. So we really want to know whether the genes that are turned on in mice and turned off in mice are the same genes that are turned on and off in human neurons. And we're hopeful and even cautiously optimistic that a lot of them will be the same, but we really need to know that in order to move forward. We also want to determine if RA1 acts as a brake on homeostatic upscaling in human neurons. And if so, is this something that we can use as a potential therapeutic strategy? Can we test if reversing upscaling in these circuits, either in developing circuits or in mature circuits, whether this improves symptoms in SMS mouse models, like the ones we've heard about tonight? We're also interested in how the chromatin landscape impacts RA1 binding and dissociation. So I showed you a model of chromatin. So these are these histone proteins with DNA, and these tails of these histone proteins can be modified by various enzymes. And this changes basically the ability of genes to be turned on and off. And so we think that in this context, that these modifications are also changing how RA1 binds to its target genes. And so we want to basically determine how these different states affect RA1 binding and ask whether we can make the states more permissive for RA binding. Because as we know, we do have one good copy of RA1 to work with. And if we can boost the function of RA1 by making this chromatin environment more permissive, this might allow some functional recovery. We can also ask whether we can bypass RA1 entirely and modify expression of specific genes by altering the landscape, again, focusing on normal downstream RA1 target genes. But this would be an approach, even if RA1 replacement didn't work, we could still target this possibly in adult circuits. And finally, we want to use cellular reprogramming to target RA1 in distinct human neuron subtypes and to look at this in patient-derived neurons. We'd like to generate neurons from patients and determine whether these phenotypes that we identify with RA1 loss are actually what we see in patients who are suffering from SMS. So let me just close by thanking again all the people that did the work, and thank you all for your attention.
Excellent. Congratulations, Mike. Those are absolutely very inspiring and very informational and educational. So I'll start with a question that we received online, again from Richard Cartwright. Is RAI something that can be synthesized? I'm ignorant of this, but curious if it is something that can be effectively absorbed, for example, a nicotine patch. Does RAI have to be delivered intravenously directly to the brain? Maybe I can quickly mention that, obviously RAI is produced by gene expression. So it's something that in theory could be synthesized. However, to bring that into the brain and absorb it is a very complex process. So certainly cannot be done through a nicotine patch. It needs to reach directly into the brain. And then, as mentioned by Mike, is the regulation and the function is largely within the nucleus. In addition to that, RAI is a very complex and large protein. So it's pharmacologically, I would say, impossible to do that through the skin or through a nicotine patch, or even intravenously will be almost impossible to go into the brain. In addition to that, due to the high instability of the protein itself, this type of approach will not work, will be very difficult to approach. What you can do, though, and that's the therapeutic intervention you heard previously, you can induce the increase of expression of the gene into the brain. You can use a gene editing and gene therapy approach, Cas9 approach, to be able to increase the gene expression. And that's one of the approaches that you heard. Or you can use a small molecule that could somewhat stabilize the expression of the gene or increase the expression of the gene, or some of the pathway, the genes that are downstream of RAI. So it's what you mentioned as an approach, unfortunately, is non-possible, too complex. But the other approaches that we discussed today in terms of gene regulation or potential small molecule to activate the gene, it's something that it could be possible. And some of the experiments that Mike and Weizhang and Jessica referred to would use potentially some of these regulators. Another question that I have actually for you, Mike, is obviously this is quite fascinating. Now, based on the knowledge and understanding of the checkpoint activity of RAI as of today, this at least suggests to me, and maybe I'm wrong, that there could be a true role of RAI throughout life, including in adulthood. So there could be a potential role in development. Therefore, it's a role that if not restored during the neuronal development, maybe there is a damage or an alteration which is not reversible. But based on what you mentioned, and if I understand correct, that was done in adult mice, so there could be a role in the adult population as well. So this gives me a little bit of a hope of thinking that if you're able to revert the activity in the adult, you might see an effect. Is that like too much of a positive of thinking at this point? Is that only speculation, or you think it's plausible that treating the adult could lead to some benefit?
So I think it's a great question, a very good question, very deep question, and something we thought a lot about. So we think that, so there's no question that to the extent that RA1's role as a homeostatic regulator, that's going to be an important role both in development and in adults. So in that, there's hope that there's an ongoing deficit that can be corrected. Now, it just so happens that developing circuits are particularly vulnerable to loss of this control and loss of this instability. So that's where you're going to see probably the biggest sort of detriment of loss of RA1. But these are things that are important throughout life. We think that these stabilization mechanisms are really important even in mature circuits, and there's actually evidence for that from a variety of sources. So insofar as this is the defect, or this is a core defect, then yeah, I think that it does offer the potential that adults could respond to treatment options if we can reverse it.
Yeah, thank
It's a
you.
great question.
Excellent, and absolutely fascinating. So I think this fantastic work. Thank you so much.
Thank
All right,
you.
I have a last question here, which is not for you. Was actually in the beginning of the discussion from Jen Goglia. The question is, what is the average age that kids are diagnosed? I have to say, I honestly don't know if there is data today on the average diagnosis, at least in the United States. What I can tell you that is largely dependent from probably country to country, state by state, the ability to do early diagnostics and genetic testing. I think there's a huge variability across the board, I'm pretty sure. What certainly we observe is that in the past, diagnosis happened very late because obviously of the fact that genetic testing in general and genetic sequencing was much more complex, expensive, and not routinely done. Today, we can do genetic testing and sequencing for a few $100s or even less, and probably it's going to be even less expensive in the future. So I do believe that it's going to happen more and more frequent. That's why we see a lot of diagnoses now within the first 1 or 2 years of life already in SMS. All right, and with that, I think today we'll learn a lot about future options. I think this is extremely high quality research that we've seen today. I am extremely pleased about the progress. As you saw, there is a progress in understanding the underlying physiopathological mechanism of the disease. There is a better way also to measure, even see visually activity of neurons. And we even saw promising potential for therapeutic intervention, including small molecule or gene therapy with CRISPR-Cas9. So I encourage further collaboration across all these different groups. And with that, I think we can conclude the discussion today.